So I think most of you know me, but for those of you who don't, I'm Susanne Rafelski. I'm Director of Assay Development in the Allen Institute for Cell Science. So we'll switch just a little bit, switch tracks a little bit away from brain and neurons to stem cells. And I'm actually here just to introduce the Institute a little bit and what our sort of big vision is. Some of this should be familiar to a lot of you, but just to get you all lined up for our awesome team talk that's coming after this. So my talk should be pretty quick, and their talk will be much longer. Okay, so why is there an Allen Institute for Cell Science? Well, that's because we actually don't understand the cells that well in the sort of basic fundamental way that the cell behaves and how it performs all of its functions. So our mission is to understand and predict cellular behaviors in normal pathological and regenerative contexts. And there's a key word there which is understand, and a key word there which is predict. And we want to do this by developing dynamic visual data on cell organization and activities, and really develop an integrated computational model of the cell that is based on the experiments that we and all of the cell biology community gathers. And so the need for this is because we've learned how to identify all of the parts in this sort of genomic era, and of course we all know that it's very hard to figure out how those parts come together. And so we really want to emphasize that the activities that drive cell behavior come from multi-scale structural specializations that are transient and localized, and that's lots of words. But the point is, the cell has many different hierarchy of organization, which I'll show you in a moment. They have to all come together. They have to all be able to do the right thing at the right place at the right time, and that's difficult. And that does not come from knowing what the parts list is. And once we sort of build this framework of understanding how the cell is organized, then we'll be able to use that to promote understanding, predict function, and provide a platform for modeling. So when I talk to you about what our first project is, that's really going to be a focus of that. Of course, our principles are similar. They should be. We want to do research that cannot be done in a typical academic environment. We want to share all of our data and tools with the research community, and everything we do is very tightly intertwined with the research community, with several communities, stem cell community, cell biology community, and so on. And of course, we have milestones and outcomes. We officially really launched our scientific activities in February of this year. We're about 35 people. We're going to be about 65, and we're very happy and proud of what we've been able to accomplish in just the last less than a year. So stay tuned, and hopefully you'll be impressed too. So at the center of everything we do is the microscope and live cell imaging, and that should be a movie that's playing, but apparently decided not to play. But that's worthly starting for, so. Good, okay. So at the center of everything that we do is the microscope. Everything we do is image-based, and I will tell you reasons for that. And that's because we want to understand spatiotemporal cell biology. We want to know what's happening again, where and when it's happening. So at the focus and center of what we do are high-resolution, dynamic 3D images of organization and activities. Now that's a pretty big, broad goal, so let's narrow it down just a little bit. We have a system, which are the human-induced pluripotent stem cells. So those are the stem cells that you can get from your skin, and they have the potential to become many different cell types. We're going to use these cells both in their undifferentiated state and in their differentiated state into cardiomyocytes as the differentiation that we picked. And we're going to, in these cells, label a whole bunch of cellular structures. And we're going to do that together with the community and make engineered lines available to the community. And you'll be hearing about the first lines that we have released just a few weeks ago. After we image these cells, and again, you'll see lots of examples of that, we're going to want to be able to build algorithms and tools and release those to the community to be able to do image analysis and feature extraction. And that will lead to models and theory. And most importantly, there will be an output that's accessible to the community to make this useful to everyone. And if there's one thing I want you to be able to remember, it's not these words, but it's that everything we do is image-based because the images hold so much of the information that we need. And in order to extract that sort of information, we need to have very high-quality images and very high replicates. This will lead us to understanding correlations in our data, lead to predictions and hypotheses that we will model, test, and the community will test as well. So that's sort of, if there's one thing, I'm going to go into 2 more slides about this. That's the one take-home message if you want to learn something about cell biology in this era. So this is the cell. You've got 4 orders of magnitude. Proteins have to find each other at the molecular level, and they have to generate activities that span hundreds of microns. And these different orders of magnitude of cell organization actually are all very dynamic and interacting with each other, and all of the components interact with each other, and that's what leads to the function and behavior. And this goes in both ways. So if you have a cell that becomes cancerous and looks like it's not behaving very well, you might be able to look at the cell and see that as an output of the dysfunction. So that's what pathologists use when they try to diagnose cancer from microscope images. But the other way around, you actually produce the function or behavior by which aspects of the cell can find each other and can perform what they need to do for the cell to function, or when that goes wrong, for the cell to not function. And that becomes even more apparent if you think about cell types. Right? Cell types and what they do, their specializations are actually just due to repurposing of the same structures over and over again. And so if you understand where those structures are, how they are composed, and how they change in relation to what the cell is doing, you can actually extract a lot of information about what the cell was doing at that time. So that leads me to why we have the first project that we have, which is the physical organization of the cell. So we want to create a physical framework for studying cell behavior and to model that behavior. It makes a lot more sense to look at where signaling events occur if we know exactly what parts are needed in those regions where the signaling occurs. So the first goal is to determine where are all of the major structures in relation to each other. And the way we do this, which you'll hear a lot more about, is to fluorescently tag each of these structures in very well-characterized normal cells, as normal as we're going to get them. Develop models for positioning and integrating these structures, which you'll hear about as well, and have a way of accessing all of that information. Then, that's when we can leap into all sorts of wonderful cell biology. How does that integrated cell, all those structures together as a unit, behave as the cell does its normal thing, as it divides, as it differentiates, and how does it respond to any perturbation that you would like to think of? Disease mutations, drugs. And that's, of course, just the start, a very powerful start. But then, we can look into signaling, we can look into cardiomyocyte-specific activities, or we can take deeper dives into, for example, the giant nuclei that these cells have, which you'll see shortly. We will definitely need to be able to assay and look for functions and behaviors in these cells, and that's going to be able to be leveraged by all of this information. We really want to integrate the omics, right? So one of these sort of big challenges in biology is to take that parts list and take the activities and put them together and understand how they fit together if you really want to understand the cell. And of course, we want to model cell behaviors, states, and types. So that's the group. So I want to thank everyone. And I wanted to point out the 4 members, the 4 scientists who are going to tell you all about what we're actually doing today. So thank you.